Hello students, welcome to this class. Now in this class we are going to learn about what is DNA and what is its structure, how does it look. Okay, so this is going to be a very useful class for all of you. So let us now delve into the structure of DNA. Okay, DNA, as you know, that it is present inside the nucleus of a cell, right? So we have a cell, and inside this cell, if you see, this is our cell, and inside this cell we have nucleus, and inside this nucleus you will see DNA. Okay, so now we are going to understand what this DNA is actually, right? For our understanding, what we say is that DNA is, DNA is your genetic material, and it transfers the information from the parents to their children, right? So how does it happen? What actually it is? How does it happen? We can learn later, but first we have to see what is DNA. Okay, let's move. So let me write the full form of DNA first. So DNA is nothing but, it stands for deoxyribose nucleic acid. Okay, now if you see, there is something called as deoxyribo part, and secondly we have nucleic acid part. Okay, so with this we are able to, we can deduce the understanding of DNA. So it has something called as deoxyribose. So what does it mean? So if we try to understand the structure of DNA, we will see that the DNA consists of three different parts. Okay, number one is your phosphate, then we have sugar part, and we have nucleotide. Okay, which means we can say nitrogenous bases. Okay, we can say this as nitrogenous bases. Now let us understand what are these structures exactly. So when we learn about DNA, we see that DNA has a phosphate group which looks something like this. Okay, so this is our phosphate group. So this is our phosphate group, and we have sugar. Now let's see how a sugar looks like. Okay, how a sugar looks like, and we have something called as deoxyribose sugar. So our ribose sugar will look something like this. This is our ribose sugar, which has one hydroxyl group here in the third position, and in the second position we have one hydrogen. Okay, and here we will be having one more hydroxyl group attached to this carbon. Okay, now if you try to balance this, you can just balance it by adding hydrogen here, one hydrogen here, one hydrogen here, because we know that the valency of carbon is 4, which means it can combine with 4 different atoms, right? So similarly our valency of oxygen is 2, so we have to see that, and then we have to proceed. So this is our sugar, and what type of sugar is this? This is called as deoxyribose sugar, and since it has a 5 carbon, so sometimes we call it as pentose sugar. Okay, which consists of 5 carbon, and ribose is the its actual name. Now in that we see that phosphate group is present. So because of this phosphate group, what happens is that now DNA can act as an acid. Why? Because in this phosphate group, the one oxygen atom which is there can bind with hydrogen. So it occurs like this. Okay, the structure occurs like this. So when in solution, what it can do is it can donate this hydrogen, which means it can just release its hydrogen into the solution, which means it is giving hydrogen. So an acid is something that gives hydrogen ion in the solution, right? Okay, now this is about acid, this is about sugar, and the rest part is our nitrogenous bases. Okay, let's go into the nitrogenous bases now. So what is nitrogenous bases? So nitrogenous bases are, so these are bases, which means it has the capacity to receive hydrogen ion. Okay, so uh, because it consists a lot of nitrogen, and nitrogen has something called as lone pair of electrons, and because of those lone pairs, it has the tendency to capture hydrogen. And when any molecule is capturing hydrogen, then which means that it is a base. Okay, so now let us see. There are if you are, we have to understand that there are four different nitrogenous bases present in the DNA. So let's see what are those. Okay, first we see that we have something called as adenine. Okay, and we have something called as guanine, then we have cytosine, then we have thymine. Let's see how does this structure look like one by one. Okay, okay, so adenine looks something like this. So this is our adenine, right? We have here ammonium group. And now so let us balance the bond here. So let me make here double bond, here double bond, and one hydrogen can come here. Okay, and all the other nitrogen, if you see, they are bonded with 3 bonds, because it has the valency of 3. Okay, now this is our adenine, and we have a similar structure of guanine, but not exactly similar. If you see, this will be something like here double bond oxygen will be there, here NH2 group will be there, and then this will be the same here. So this is our guanine, and here one hydrogen can come, here one hydrogen can come, and here one double bond you can put. So similarly, if you see cytosine and thymine, so cytosine, so if you see these two structure are having 2 rings, right? So 2 rings are there, so that's why we call this as purines. Okay, these are also called as purines. Now cytosine and thymine only has one ring structures, so they are called as pyrimidines. Okay, let's see how this 2 pyrimidines looks like. Okay, so similar to, if you see, this is also similar to adenine, but here oxygen is present in this position. Okay, and here double bond is there, and wherever we need double bond we have to make it. Now this is how our cytosine will look like, and the thymine will look something like this. Okay, here double bond, and here one methyl group. So this is your pyrimidine. Now how does this DNA is arranged into, all these things are arranged into a structure called as DNA. How does that happen? Let us see in the next slide. Now let us see the structural arrangement of DNA. So when one, now I can draw it like this also, so that I can draw it faster. Okay, now if this is our sugar unit, okay, and here we have a nitrogenous base. Let me see this, write this as NB. So this is my nitrogenous base. Okay, let me draw it little neatly. Nitrogenous base. Then here we have phosphate group. So carbon will be there. So I need not draw C here. I can just put it like this. Then phosphate group I can just add like this. Okay. So now this phosphate is there. This forms the backbone of DNA. So DNA, this particular part which you see here, this forms the backbone of DNA. Backbone of DNA. And to complete this, let me draw one OH here. Okay. Then O like this. Okay. Now this forms the backbone of DNA, and this particular part which forms the backbone of DNA along with the nitrogenous bases, or everything put together, is called as, we call it nucleotide. So what is a nucleotide? Nucleotide is the unit of structural unit of DNA. Okay, similar nucleotides will be repeating again and again and again to form a chain of DNA, and one more chain will come to make a double helical structure of DNA. Okay, so we learned that there are four nitrogenous bases, right? Adenine, we saw guanine, thymine, and cytosine. So if you see the pairing of these three, these four, how does the pairing occur? So A pairs with T with a double bond, G pairs with C with a triple bond, T pairs with A with a double bond, and C pairs with G. So here if you see that A always pairs with T and G always pairs with C. So what you can, how can you remember this? You can just remember one sentence. You can just remember, at government college. So if you remember, at government college. So at means 80 pairs, government college means GC pairs. So this could be used for remembering the structure of DNA. Okay. So I have explained you the basic part of DNA and how does it look. Now let us now actually go and see a little bigger structure of DNA. So how does a bigger structure of DNA looks like? So let us see that this is the nucleoside. Okay, now I'm simply drawing a line here. Let me say that this is a line. Okay, and this line is what? This line is your sugar phosphate backbone. You can see this way, sugar phosphate backbone. If you see in the previous one, we see that this is our backbone, right? Here phosphate is there and ribose sugar is there. So this ribose sugar and phosphate gives you the uh, sugar phosphate backbone. Now let us move back and then see that. Let me highlight this little more here. So let me make it a little bigger. Okay, this is my sugar phosphate backbone. This is how my DNA will run. Then now in between what we have is, we have a nitrogenous bases. So different bases are present, right? Adenine is present and it binds with thymine with a double bond. Okay, so we can draw it like this. Then T A. Okay, A binds with T. Similarly we can make G C A T G. Okay, C G T A C. So this, don't you think this looks like a ladder? So we call this as, the nucleotide bases are the, looks like a rungs of the ladder. Okay, this part, this is our, looks like rungs, right? and this sugar phosphate backbone looks like the side support of the ladder. Okay, now how does this actually in a 3D space, how does it look like? It is not a simple linear structure, but DNA is a plane. Okay, DNA is a helical structure. So if I have to draw a helical structure, let me draw it here. So helical structure will look something like this. Okay, yes. So in the between we have these bases attached like this. Okay, so bases are attached like this. So this is our structure of DNA, that we have a double helical structure which is present in inside our body like this. And this line which I have drawn is the sugar phosphate backbone, and the middle part is the pairing of nitrogenous bases. Okay, so with this we come to the end of the structure of DNA. Just to quickly give you a brief idea of how actually one particular nucleotide pairs with another nucleotide, let's just have a look at that and we close this session. Okay, so now this is my nitrogenous phosphate group here, and I have, let me draw one sugar group here. Okay, this is my sugar group, this is my phosphate group, and this is my nitrogenous base, and I have one more nitrogenous base in this same way, and this nitrogenous base is attached to, let me draw the sugar in the opposite orientation. Okay, so if I want to draw this sugar in the opposite orientation, then we have oxygen here, then the oxygen will bind here, then another will come here, then here, something like this. Okay, and with this we have one phosphate bond, phosphate. Okay, so if you see here, on one side the orientation is different, right? Here orientation is this way, and the on the other side orientation is different. So now another nucleotide will come, and that nucleotide will bind with the phosphate here, with this phosphate. Okay, here it binds with that, and which part of that nucleotide will bind now? If I were to draw one more here, so if you see here, it will bind like this. Here we have the phosphate, and here if you see, one oxygen was there, right? So this is how it binds. Okay, here. So, and here similarly we have nitrogenous base, and if you see here, hydrogen bonding will be there, and another nitrogenous base, and same structure here. Okay, here one phosphate, and this phosphate will be connected to the, here one oxygen will be there, so this oxygen, it will be connected to this oxygen here. So this way our DNA structure will be repeated again and again. Here our, based on bases, double or triple bond will be there. So what is this part? This is called as hydrogen bond. Okay, this is also your hydrogen bond. So this is the structure of DNA, which you must learn before proceeding further, so that you have a very clear understanding of the structure of DNA. So let us meet in the next class, where we can learn about the chromosomes and how a chromosome looks like, we can just have an understanding of chromosomes in the next class, an entire picture including what is chromosome, how does chromosome look like in meiosis, what are homologous chromosomes, what are chromatids, sister chromatids, non-sister chromatids, dyad, tetrad, all this we can discuss in the next